So in today's class, I will be talking about cell cytoskeleton. Uh, Before we start cell cytoskeleton, just know that like we have a skeleton in our body made up of bones, our each cell also has got skeletons, and these skeletons are made up of three proteinaceous filaments, and they are known as microtubules, microfilaments, and intermediate filaments. So three types of filaments, they make the skeleton of the cell. And as you see, they are attached to the plasma membrane. They are associated with plasma membrane, and they are associated with different organelles and all. So they serve not only to give support, but they also act as roads on which several types of transport within the cell takes place. Maybe a vesicle moves from one side to the other. Maybe something walks from one side to some protein walks from one side to the other. So these are the different functions which you can imagine these microtubules to be taking place. Okay. So with this, I will share my slide. So essentially, like I mentioned, that the cytoskeleton, it's the skeleton of the cytoplasm. And you can see this is a section of the cell. What you're seeing, just a moment, I'll put the. So I'll increase the size a bit so that you can see it properly. So I hope you can see this. You have the plasma membrane, which is covering the cell. Inside the plasma membrane, you have one type of filament known as the microfilaments. These brown fibers which you are seeing, which are coming down, brown colored fibers. This is the cell mitochondria here. Then you have these pink colored filaments, these filaments which you see, they are known as the intermediate filaments. And you have these thick filaments over here. These are known as microtubules. On top of the microtubules, you find vesicles present. So with microfilaments, mitochondria, and intermediate filaments, microfilaments, microtubule, and intermediate filaments, they together make the cytoskeleton of the cell. Now, what is the function? So the first function is structural support. So it gives a structural support, and it maintains shape of the cell. The shape is there, and it provides anchorage for organelles. So the different organelles like mitochondria, Golgi bodies, which are present inside this cytoskeleton, they provide an anchorage. They hold on to these organelles. Otherwise, the organelles will all fall down in the bottom of the cell, so that they are held throughout, suspended throughout the cells. It's held by this cytoskeleton. It helps in motility, movement, like cilia and flagella, the microtubules. They are useful for locomotion, beating of cilia and beating of flagella. And it is also important in regulation, organizes structures and activities of the cell. One important activity of the cell is movement of substances from one corner to the other corner. Normally they may occur through vesicles. So these vesicles, they move through these microfilament fibers, or the microtubules, or these cytoskeleton fibers. So basically, you can find three main functions. The first function is structure, movement, and regulation of cell activities. So cytoskeleton has got three types of function. So if you see, it is basically a network of fibers extending throughout the cytoplasm. This is an electron micrograph. You see, it's just a network of fibers going throughout the cytoplasm. There are three main protein fibers. Uh, that is the microtubules, actin filaments, microfilaments, and the intermediate filaments. So basically, this classification is on the basis of the size. So have a look at this. This is a cross-section picture. You have the plasma membrane on the top, this purple color, and the plasma membrane on the bottom. Then these green colored fibers, they are the intermediate filaments. Then these little fibers, these lines which you see going here, they are the microfilaments. And these blue lines you see below, they are the microtubules. So you can see that how the mitochondria is attached to these intermediary filaments, how they hold this filament to, how these intermediate filaments, they hold the mitochondria together. This green network, it's holding the mitochondria. So in the cell, they are basically supported in this manner. So from the evolutionary perspective, you can see that proteins that make up the fibers are very similar in all living things, from bacteria to humans. So they are very similar. Tubulin is found, for example, tubulin is found in all cells. Actin is found in all eukaryotic cells. This means that they are both essential in the ancient as well as modern organisms, and they are very essential for life. So first, let us talk about the microtubules. We are going to talk about the microtubules. So if you have a look, this is a structure of a microtubule. They are the thickest fibers. They have got a 25 nanometer diameter. They are constructed of a protein known as tubulin. This is a tubulin dimer. Tubulin exists as a dimer. One is this deep yellow one, and one is this light yellow one. They are alternating. You see one line of deep yellow, one line of yellow, this light yellow. So basically, two dimers, they join together and form this tubulin dimer, and they form this tubulin filament. So they grow or shrink as more tubulin molecules are added or removed. So when this tubulin dimer is added, tubulin dimer, one here beside another one. So when this tubulin dimer is added, They keep on adding, and they become form a. But as you know, the tubulin microtubules are the most important tubulin. So, what is the function? Structural support and cell movement. They move chromosomes during cell division, spindle fibers found present in centrioles. They have got another function, that is they track, guide motor proteins carrying organelles to the destination, like kinesin, dynein. So they act as a track. This is the tubulin fiber. This is acting as a track. This yellow color you're seeing, microtubule of cytoskeleton. On top, this is an organelle, and this is this is a motor protein. This is the motor protein here. Okay, by hydrolyzing ATP with energy on top of this, this is a road on which the car containing the organelle is moving from one side of the cell to the other. It also helps in motility, like in cilia and flagella. In centrioles, you will see spindle fibers coming, astral rays coming out from there. This spindle fiber comes out from structures known as centrosomes. In the centrosomes, this is a centrosome, which you see. Inside the centrosomes, there is a pair of centriole. This is one centriole. And this is the other centriole. From these centrioles, centrosomes, you have spindle fibers and astral rays coming out. Astral rays are spindle fibers. So what are cilia flagella? Cilia flagella, they are nothing. They are only an extension of the eukaryotic cytoskeleton. They go outside the cell. For example, look in sperms, you can see this tail of the sperm. This is this flagella of the sperm. This is basically an extension of the cytoskeleton. So cilia are numerous short hair-like, and flagella are 1 to cell longer whip-like. They move unicellular and multicellular organisms by what flagella does, by propelling water past them. What cilia do is they sweep mucus and debris from the lungs. You know how paramecium walks with the help of cilia, flagellum of sperms. So cilia have got an oar-like movement. In a boat, when you row, you have these two wooden rods. They are known as oars. So they move in that manner, up and down circular motion on the water, and they move like this, and this helps in the direction. So active stroke from here to here, this is the active stroke which takes place, moves from original to this position, and the recovery, it comes on like this. See, from this position it goes to this position, from this position it goes to this position to this position, and back it goes to recovery. Like you are swimming, you are giving an active stroke like this, and then you are gradually recovering it out. So this is how cilia work. Flagella undulation. Undulation means circular movement. The flagella goes like this. Like this. In this manner it goes. So this is a undulation movement. And this causes the euglena or the sperms to move ahead. This is a sperm which you are seeing. Now we have cilia and flagella structure. They have got 9+2 orientations. So what is 9+2 orientation? If you see, this is 2. This is 1. This set is 2. This set is 3. This set is 4, 5, 6, 7, 8, 9. And in the center also there is 2. So you have 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 plus 1 and 2. 9 plus 2 orientation. This is what you mean by 9 plus 4. Each of these are microtubules. So two together they form a microtubule. So 9 plus 2 it makes a flagella. But flagella is made up of many 9 plus 2 microtubules. That is around 11. So bending of cilia and flagella is driven by a motor protein. The protein moves on top of this. As you can see, let's increase the size of this figure. So in between these microtubules, you see there are these little arms, dynein arms. So what they do is they slide, they exert pressure. So you have it's something like this. So when you it moves, there is a pressure. So this twists and turns. From here, it takes a movement and it becomes, say, here. So this sliding movement, it causes the microflagella to bend. The flagella to bend or the tubulin to microtubules to bend. So this is the same structure. This is the central microtubules, 9 plus 2 orientation. There are triplets also found somewhere. Now we go to actin filaments. They were formerly known as microfilaments. Now they are known as actin filaments. They are the thinnest class of fibers. In muscle contraction, you have contraction of actin on top of myosin arms causes a muscle to contract. So these actin are very important elements in muscle contraction or cell contraction, cell movement, cell structure, cell shape. Actins have got a very important role. It's like a coiled protein like this. These little units are coiled. 7 nanometer. They are the thinnest. They are solid rods of protein. Twisted double chain of actin subunits. So each the circle is an actin subunit. Together they form an actin filament. 7 nanometer in diameter. It forms a 3D network inside the cell membrane. In muscle cells, actin filaments interact with myosin filaments to create muscle contraction. I have mentioned before. So it is a dynamic process. Actin filaments constantly form and dissolve, making the cytoplasm liquid or stiff during movement. So when you study locomotion in amoeba, you see these, you have outside region of the cell, gel-like actin network that is known as gel. And these actin dimers, they dissolve and become solubilize and become sol, these little dots. The moment it becomes sol, it becomes liquid. And the moment it polymerizes and forms actin filament, outside is a jelly-like structure, solidish. Then we come to the intermediate filaments. They have a special structure. They are specialized for bearing tension, pull and push. Especially your hair protein, skin protein, keratin. Same protein as hair. Intermediate in size, that is 8 to 12 nanometer. So it looks like this, fibrous. They hold things in place inside the cell. More permanent fixtures of cytoplasm. They reinforce cell shape and fix organelle location. The nucleus is held in place by a network of intermediate filaments. So in a summary, the microtubules are thickest, cell structure and motility. There you have tubulin, Actin filaments, they are the thinnest. Internal movements within cell, actin, myosin. Intermediate filaments, intermediate, more permanent fixtures, keratin. So this ends my class here today. Only 12 students have attended. Very poor attendance. All the names are reflected over here. I don't need anything else. So do you people have got any questions? প্রশ্ন থাকলে বলো। তোমরা আমাকে শুনতে পাচ্ছ? হ্যালো?
Yes, sir.
Okay. So do you have any questions?
No, sir.
No questions. Okay, then I will. Asters and spindle fibers. What is the difference between asters and spindle fibers? This is one question. There is no difference between asters and spindle fibers. They are the same. The astral rays, they extend and they become spindle fibers. They are the same thing. Aster means you have seen an aster flower. It's like this. 1, 2, 3, they are coming out. But once when they come out, the fiber is a spindle fiber. So this is astral rays is near the centrioles. You can see, yeah, it's like rays coming out, like an aster flower. Therefore, they are called astral rays. But they are eventually. This is the difference. That the location near the microtubules, that is the astral rays. But then in the center, where the microtubules holds the two centrioles on both sides, centrosomes on both sides, then that's the spindle fiber which holds the chromosome. There is no difference. That's a good question. At least that's a good doubt clearing question. This is how. So I end my class over here. Any more questions? Please do not hesitate to ask. And you people, let me tell you one thing. I am from the career counseling group. You people are apply for these EduBridge, Humansys, TCS, NQT. Even you're in the second year. This is valid for 2 years. The day you pass out from BSc, you will be in line for a job. Join it. It's a very cheap rate. All these things are taking place. 2 years courses, 3 years courses. Even the people from biology get proper jobs over there. I can tell you, promise you one thing: you will not get government jobs. Whether you go to police, post office, railway, you will get jobs only for sweepers, construction workers, like Principal Sir told. Those jobs are there. But after studying, doing a sweeper's job, well, I think people can do better than that. So seriously apply for these. If you have doubts, ask me. Don't le are 2 saal baki hai, don't go. Humansys abar ashbe, provided we can give them 100 students. Therefore I'm saying, but now you can join EduBridge. That's a good company. Then in November, there is a TCS NQT. The score is valid for 2 years. Lots of biotechnology, life science companies are also associated with TCS NQT qualifiers. Right. And so with this score over there, you can keep on applying for jobs in TCS NQT. Don't waste your time. Start right now. 2 years will go like this. उसके बाद नौकरी नहीं मिलेगा. No government jobs. Let me. I can promise you that. I never seen I've never seen maybe one student only. I've seen in the whole zoology department, only one student got a government job. The rest are still preparing for last 5 years. They're preparing for competitive examinations, wasting their life. They have crossed 25. They're preparing for competitive examinations, keeping on question. So it makes no sense. Don't become stupid like this. And I can tell you, they were much better students than what you are. They were very, very good students. It's not that they were faltu students. They were very good students. With the current batch, I cannot speak proudly of my current batch, but with my up to 2019, all those students, Whenever I think about them, I see they're high quality students. I speak about them proudly. They are going abroad here, there, JNU, BHU. They're studying all over India, all over the world. They're doing very well. From last 2 years, heavens knows, from 2019 batch, what happened? All interest of the students are gone, and we are really frustrated. So I would say tell you people to be very serious about their career. Quality of a student is actually not the brain which you have in your head. It is your attitude that I want to do something. मुझे कुछ करना है. Whether you are good or bad, that is what defines. If your ambition is always कि मुझे रेलवे प्लेटफॉर्म गवर्नमेंट जॉब में झाड़ू देना है, well, your ambition is that is your attitude. But if your attitude is that I want to be a top research scientist in University of Edinburgh, that is also your attitude. That's a positive attitude. You're thinking high. Maybe you will not be able to reach Edinburgh, but you will definitely reach somewhere around some Dundee or Aberdeen University in UK. You will definitely reach there. This is a promise. This I've seen. It's your attitude. मुझे कुछ करना है. I've seen it. Very soon I will invite one of my students. He passed out in 2019, 2016 batch entry, first CBCS batch. Sanjay Das. His father used to sell chow mein roll on footpath, and his brother used to drive a Toto. He used to study in our department. He scored. He not only scored highest, all the teachers after taking Viva from different colleges, still they ask, what is Sanjay Das doing? He was a great student. What a Viva he has given. His concepts were very clear. Teachers kept on asking him tough questions. He kept on answering them conceptually. Till the teachers had to give hats off, and he got full marks, 40 out of 40 in molecular biology. He cracked national eligibility test. He cracked CSIR national eligibility test, and he's right now finishing his MSc in JNU. And right now he's got selected as a PhD research scholar in two places, one in Max Planck in Germany, one in Switzerland, EMBL in Switzerland, top institutes. His father from Asansol used to sell chow mein and roll on the footpath. So people from these background also, they go to this success level. With a 2 megapixel camera wala phone, only so I saw the RAM was 750 MB RAM. With that he studied all the top textbooks. So I had students like this. So this is because of his attitude, his background. He's the first generation learner. His parents did not know, just maybe they know how to sign their names. That's all. And he's going on to this level. So and he's just a simple street guy. One day I was walking through SB Gorai Road, one lane over in SB Gorai Road, and suddenly I saw that he was serving chow mein to some people from a roadside stall. Then I asked him, sir, this is my father's stall. I am helping him out today. I got to know that his father is a chow mein seller. Then one day a telephone came. Then my Truecaller showed Chow Kanuda. So his father was Kanuda, and chow mein, somebody saved this number, and from Truecaller I got this. I came to know he was calling from his father's phone. So from these I got to know about these things. So you see, it's attitude which makes a person, not your background. Also has got an effect, but still, you people are studying in one of the best colleges in this region. Your background should not be bad. If you take advantage of this college is going to give you good benefits in your career. You take advantage of the career council group. Okay? ঠিক আছে, রাখছি. Bye bye.